In the final part of this week's lecture, we look at an application of induced pluripotent stem cells in the context of Alzheimer's disease. So what we've seen so far today is that neurological and psychiatric illnesses are genetically complex. And because of their location in the brain, they're very, very difficult to study from a molecular and a genetic point of view. We've seen that induced pluripotent stem cells are a way of taking an adult differentiated cell and de-differentiating it back into a pluripotent stem cell. What the final part of today's lecture does is show how we can utilize that to study the neurons of a patient carrying a mutation that is associated with Alzheimer's disease. This is the paper on which it's based, and we can see through the title that it's very clear what it's about. So it's modeling Alzheimer's using iPSCs, and it reveals stress phenotypes associated with intracellular A beta. So the formation and accumulation of these A beta oligomers is presumed to play a role in the pathogenesis and clinical symptoms of AD. We've already talked about this in the context of early onset Alzheimer's disease. Mutations in APP and the presenilins lead to an increased amount of the A beta 42 toxic protein. And we also have heard that the ApoE4 allele is thought to lead to errors in the accumulation and removal of the A beta peptide, which causes the plaques associated with Alzheimer's disease. So there are a number of mutations linked to familial AD, as I've just said, the APP and presenilins, for example. And recently, an atypical early onset familial Alzheimer's disease, which is caused by a mutation called E693 delta. What that means is it's the deletion of a glutamine, and it's an APP producing variant, and it's from a Japanese family. So what happens is that patients who carry this particular mutation, they show early onset symptoms of Alzheimer's disease, but lack A beta deposition. So what they've done is they want to understand how this mutation causes Alzheimer's disease. So what you see in this photo here is starting up at the top here, we're looking at fluorescent images through a microscope such as this, in which we see the cell culture. So what we've done is we've taken the fibroblasts of someone who has this particular mutation. For example, here it is here, APP E693 delta. So an individual carrying this mutation, there's a sample of their skin was taken, fibroblasts were isolated, and using the vectors that we talked about in the previous lecture, the transcription factors MYC, KLF4, SOX2, and OCT4 were introduced into these fibroblasts. Following treatment with morphogens, we see epigenetic reprogramming and the reversion to a stem cell through the expression of these stem cell markers called NANOG and TRA160. It's not really important what they do. Suffice to say that we only see these proteins in stem cells. What they then did was they took these stem cells and using the appropriate morphogens, they have now created differentiated neurons. And this is exactly what you can see here. So now we're looking in these fluorescent images. You can see these sort of tangle-like things here. These are actually the neurons. These are neuronal cultures, and you can see they're derived from the particular mutation we're interested in, E693 delta, as well as a second mutation that's a valine 717 leucine, as well as a sample from sporadic Alzheimer's disease and a control culture from someone who does not have Alzheimer's disease. When they culture these neurons, what they see is the following. They see that, so let's just orient ourselves about what we're looking at. So we're seeing in the top, we're seeing in green, a stain for a protein called NU1. Now, NU1 This is an antibody that specifically recognizes A-beta oligomers. Now, if you remember, I said that this particular mutation, it causes early onset Alzheimer's disease, but an unusual feature is that there is no deposition of A-beta plaques. However, what they find is the following. So now we're looking at cultured neurons. These neurons are derived from adult fibroblasts. The fibroblasts were reprogrammed using these combination of stem cell transcription factors, and then using specific morphogens, they were then directed to form neurons. So here's the control, and you can see that it's pretty well just a black background. Here is our APP E693 delta mutation, and you can see all of this green fluorescent lighting up here. What that's picking up is this new one protein, A-beta. In other words, the cells are full of A-beta peptide, and that's what you can see on this graph down here. So we're looking at the area or the amount of NU1 in the different cell types. There's several different cell lines generated in each case, and here is our E693 delta mutation, and you can see by the height of these bar graphs that these cells here are full of A-beta peptide. When you treat the cells with a gamma secretase inhibitor, remember the PSEN1, PSEN2, how they inhibit the cleavage of APP into A-beta, we can see the disappearance of the A-beta peptide from the cells. So what are we seeing? We're seeing that we're able to take a skin cell from someone who has a mutation that causes familial Alzheimer's. Of course, because it is an inherited familial disease, they will carry that mutation in every cell in their body. So taking that skin cell, it was de-differentiated into a pluripotent stem cell, and it was then redirected to form a neuron. Using an antibody against A-beta, the authors found that this particular mutation causes an accumulation in the cells of A-beta protein. They then did a gene expression study, and what we're looking at here is something called a heat map. A heat map basically says that all those genes that are expressed at very high levels will code as red, and all of those genes that are expressed as very low will code as green. So it's a very convenient way of picturing patterns of gene expression. So what can we see? Here is our control neuronal cell lines. There are 3 of them. They've been coded differently. And here is our E693 delta neuronal cell lines derived from the induced pluripotent stem cells. And what you can see is very opposite patterns of gene expression. When we look along the rows going this way, each and every row as we move up is a gene. And of course, the columns are the 3 different cell types in each case. So we see that in the control, we have a group of genes which are highly expressed, whereas in the mutation cell, those genes are turned off. And the opposite is case in the mutation. So we're seeing opposite patterns of gene expression. Now, what's interesting is that in the case of the cell line carrying the mutation causing familial Alzheimer's, these genes are responsible for what is called cellular stress. Now, cellular stress is a response to the cells, often when they undergo metabolic change or they're forced to make more protein, that can stress the cell. Their endoplasmic reticulum is unable to keep up with the demand, and what the cells do is that they slowly shut themselves down. They shut themselves down until the stress has been removed, and if that doesn't happen, what happens is they then die. So these cells are in cellular stress. And if you remember, they were also full of the A-beta peptide. So it's entirely possible then that this mutation causes an accumulation of intracellular protein, A-beta. This A-beta protein causes cellular stress, as seen by this gene expression study here. Now, what's interesting is that there is a type of acid called docosahexaenoic acid, or DHA for short. Now, it's an abundant long-chain polyunsaturated omega-3 fatty acid in the brain. and it's highly enriched in synaptic fractions. However, the level of this omega-3 fatty acid is decreased in the brains of Alzheimer's patients. Therefore, it's been suggested it may be one therapeutic method to improve the symptoms of Alzheimer's by increasing the level of this particular omega-3 fatty acid. What they found is that omega-3 fatty acid consumption reduces Alzheimer's disease risk, and DHA modifies the expression of Alzheimer-like brain pathology in mouse models. Unfortunately, however, giving DHA to a cohort of Alzheimer's patients showed no difference in cognitive decline over the study period. In other words, there is an omega-3 fatty acid, highly enriched in the brain, particularly in synaptic fractions, decreased in Alzheimer's patients. Therefore, a trial was conducted to increase the level of this particular omega-3 fatty acid in the brains of Alzheimer's patients. However, this trial failed, which brings us back to our iPSCs and our particular mutation called E693 delta. So what the authors have done is the following. First of all, the columns in blue are our control neuronal cultures not carrying a mutation, and the columns in yellow are our E693 delta familial Alzheimer's cultures derived from our iPSCs. Here is the untreated control, which is DMSO. This is the level of intracellular A-beta peptide. And there are also measures of cellular stress as well. So what happens? Here is increasing DHA, and what you can see is that in our E693 mutations, that in each case, we start to see a decrease in this level. Here is a plot again, that with 5 micromolar DHA, up to 16 days, we start to see the increased survival of these particular neurons. And that can be seen when we compare it to our control. So for example, here is our control, and you can see that in the absence of DHA, the death of the neurons is very steep, whereas as soon as we add DHA, after 16 days, we see a slowing of neuronal death. So what it's saying is that at least in these neuronal cultures that are full of the A-beta peptide, that treating with DHA, this omega-3 fatty acid, that it slows neuronal death, that it improves the health of these neurons that were derived from cells of individuals with familial Alzheimer's. So it's saying that DHA increases the survival of neurons derived from the fibroblasts of an individual with Alzheimer's disease who carries this particular mutation. It suggests that indeed this DHA therapy could alleviate the symptoms, not cure, but alleviate the symptoms, and be most effective in patients carrying this mutation. Which leads the authors to conclude that patient-specific iPSCs would provide disease pathogenesis, irrespective of the disease being a familial or sporadic form, as well as enable the evaluation of a drug and patient classification of Alzheimer's disease. So what they're saying is that for the first time, we have the ability to study the effect of specific genetic mutations, or perhaps a combination of risk alleles in a culture of neurons derived from a patient who has Alzheimer's disease. So what we've seen then is Alzheimer's disease. It's the most common form of late life mental failure, counts for an estimated 60 to 80% of dementia cases. The generation of amyloid beta peptides of either 40 or 42 amino acids from the APP, amyloid precursor protein, and their subsequent aggregation and deposition as senile plaques is one of the defining events in the pathogenesis of Alzheimer's disease. The use of these iPSCs provides the opportunity to investigate the molecular genetics of neurological diseases such as Alzheimer's disease.